Philip Gingerich is not only one of the world's most distinguished archaeologists and paleontologists, he is a dear friend and colleague from Michigan. I'm so grateful he made the trip out here. It wasn't just for the sunshine. He's very eager to meet more people here. And I'm so glad he came also because his topic today has a deep relevance to evolutionary medicine. One of the pleasures of getting to introduce your friends is you get to read their CVs like you've never read them before. I never knew he spent a couple of years teaching kids science in Malawi before he began the rest of his scientific career. I'll ask him about that later. He got his training at Princeton and at Yale, and really has spent his entire career at the University of Michigan, where he's renowned for his research on whales, and especially on the evolution of whale appendages, especially the rear legs and the hip joints, of which he is the world's expert. Wonderful Smithsonian exhibit and PBS program based on that work. Also looking at his CV, I actually got the details on the awards that I've heard him getting over the years. In particular, I knew he got the Henry Russell Award from the University of Michigan, which is the award given to the single most distinguished faculty member in the university. He's been president of the Paleontological Society. He's a member of the AAAS. He's a member of, or fellow. He's an elected member of the American Academy. Most dramatically, recently in the last 2 years, he won the Romer Simpson Medal from the Society of British Paleontology, the greatest honor of geologists in his position. Yeah. In the midst of all this work on whales, however, he's had a deeply theoretical and mathematical interest in trying to figure out how we calculate rates of selection, an extremely controversial topic that goes right to the heart of debates about punctuated equilibrium and all the rest. Without taking another minute, Philip.
Thank you. Thank you, Randy. Well, it's indeed a pleasure to leave snowy Michigan and come here. It's hard to believe how you people live. I often talk about whale evolution, and I can't start today without mentioning that a colleague from the beginning of my studies of whale evolution, 1973 in Pakistan, is here in the audience, Mahmoud Raza, Dr. Mahmoud Raza. And so it's a special honor to have you here. I like to tease biologists by asking two questions. And the first is, how fast is evolution? And I usually get a lot of blank faces because a lot of people never thought about that. How fast is evolution? And I mean put a number on it. Now, if I ask you now, maybe you did the reading, and maybe you can give me the number. Anybody? Because I want you to leave with a number in your mind about how fast it is, and I want you to leave with an appreciation of what that means. Now, the story really starts with the origin of species. And it starts with Darwin saying, I do believe that natural selection will always act very slowly, I underlined that, at long intervals of time on very few inhabitants of a region at a time. I further believe that this very slow, intermittent action of natural selection accords with what geology tells us of the rate and manner of evolutionary change. What did he know about how fast evolution is? He didn't know anything about how fast evolution is. He argued that it's slow because his experience was geological and paleontological. And he argued that it's slow to make it palatable, I think, and that's my interpretation. That part of his argument that evolution is slow, and part of his argument is that it just acts on a few inhabitants of a region at a given time, is just to make it palatable. And there aren't many areas where I would imagine Darwin to have been wrong, but this is one of them. Because I think it's fast, and I'll explain what I mean by that, and I think it acts on everyone all the time. So it might have been important politically to say what he did at the time, but I think that's an area where he's underestimated what he was talking about. Now the second question, you didn't do very well on the first question. The second question is, how many figures, illustrations, did Darwin publish in The Origin of Species?
One.
I've heard a couple ones.
I'd say about
10, 15. It was really embarrassing. When I was an assistant professor, I gave a lecture at Harvard. Steve Gould was in the front row along with Ernst Mayr and others. And Steve said 2. How do you answer that? Because there is only one. There is only one, and that's this one. There are later editions where additional figures were added by editors, but the only one in the first 6 editions, I mean in the original editions, is this one. And it's a picture, it's the only picture we have of how Darwin imagined in his mind that evolution, what evolution looks like. And I want to call attention to the axes. First of all, he's got time on the vertical axis. That's the independent variable in evolution. And he's got form, phenotype, on an unscaled axis here. Here he talks about this being thousands of generations. So he scaled that axis. This one he just represented as form very vaguely. And obviously it needs to be scaled, and we'll talk a little bit about how to scale it. And I'll just say here, standard deviations are the way to scale it, or Melanobus D in a multivariate sense. And we'll see some examples of that. So it can be scaled. Why time on the vertical axis? All of the diagrams I'm going to show, except one today, have time on the vertical axis. A physicist always puts it, the independent variable on the abscissa, on the horizontal axis, on the x-axis. Why did Darwin put time running up the page?
because it corresponds to geology.
Yeah. Darwin was very much in his early days a geologist and a paleontologist, and for a geologist and paleontologist, time stacks up vertically. Now, this is how I think about the world. This is how I think about its history. This is how I think about the history of life. To me, time stacks up vertically, and that's because I'm a paleontologist. If you want to draw it the other way, that's fine too. But I'll stack it up vertically. Now, when I say what is the independent variable, I already told you it's time. What does that mean to be the independent variable? Is time a product of form, or is form a consequence, a product of time? And I think we understand that time goes by and things change through time, and therefore time is the independent thing that we can't control, that operates independently. So keep that in mind as we work through these vertical axes. Now, what are rates in this context? Here's a lineage starting at the letter E here and just going straight up the page, never changing. That I'll call zero rate. Nothing changed. Nothing changed through time. Here's a lineage that started at A and worked its way up the chart going to the right all the time, and we generally call that a positive change, and we measure it as the angle or the divergence from vertical in a positive direction, or it could equally be in a negative direction, and then we give it a negative sign, but the two mean the same thing. So when I'm talking about rates, I'm just talking about an observation at a given time, an observation at a later time. What is the difference and how much time went by? Now right away you see a rate is a ratio. Etymologically the two have the same root, rate, ratio. And we're all cautioned to be careful about ratios because they're slippery. Statistically they're slippery. And that's because numerator changes, but so does the denominator sometimes. And so what a ratio means can't be determined just from the ratio itself. And that turns out to be the same with rates, including rates of evolution. Now, I've sort of put this in a context of then and a context of now. And then is what went before, and like everything, I'm standing on the shoulders of giants, and if I can see a little farther, it's just because I'm standing on what they started. George Simpson was one of the first. He wasn't the first. A German named Schwarzkopf was the first, but Simpson was one of the first in the major features of evolution to quantify, or in tempo and mode of evolution, to quantify evolutionary rates. And he talked about the size of cusps on teeth of horses through time. Now, he didn't have a good way to quantify that. J.B.S. Haldane, a famous geneticist, proposed a unit in factors of E per million years that he called a Darwin. Now, it turned out that was a crazy number, so he wound up talking about milli-Darwins all the time. Does a factor of E per million years speak to you? I've studied this for a long time, and it still doesn't speak to me. I know what E is. I know what natural logs are. It doesn't help. So I'm going to propose another unit that I'm going to name for the esteemed Haldane here, and I'm going to call it the Haldane, but it's going to be a unit Haldane also proposed to sort of at the end of his article, after he named the Darwin, he also proposed, well, maybe these rates should be in standard deviations per generation. And it turns out that's the only practical way to measure, to quantify evolutionary rates. Now, Steve Gould comes into this because when I was young, he invented something he called punctuated equilibria, which was the idea that species can't change except when they do, and when they do, we can't understand it. And that made me crazy. I first heard of this as a graduate student. I visited the American Museum of Natural History. I met Niles Eldridge, and a colleague was talking to him about punctuated equilibria. And on the way back to New Haven, I said, "What were you guys talking about?" Punctuated equilibria. And my friend Peter Dodson explained. I thought, that's the craziest thing I ever heard. This is before the paper was published about it. I went down in the basement of the Peabody Museum at Yale, and I started measuring Hyopsodus, which is a very common Eocene mammal known from many successive stratigraphic levels. And I plotted out what Hyopsodus did through time and made a quite coherent pattern that Nature published. And of course, I struggled with Steve Gould from then on because he didn't like anybody questioning him. So I'll come back to punctuated equilibria because I think we can resolve punctuated patterns perfectly well in the context of what we think of as ordinary Darwinian gradual evolution. Now, there's some other interesting things to say about rates. Stanley Garn I've used as an example here because he was at Michigan, a very famous human biologist, and he liked to talk about the human secular trend, the idea that we're all getting taller through time, have been for generations. And I said, is that evolution? And he said, no, it can't be evolution, it's too fast. And so we come right back to the question I started with, well, how fast is evolution? You can't say it isn't evolution if you don't know how fast evolution can be. And Garn wasn't quantifying this. Second person I'll pick on here a little bit is Douglas Falconer. And I only put him up as a representative of geneticists who do lab experiments, elegant lab experiments, in his case, selection experiments on mice, body size in mice, for years and years and years. And he was nice enough, he's dead now, but he was nice enough about 15, 20 years ago to send me all his original measurements of mouse body weights, generation by generation by generation. And those rates are all plotted here. But I put this up now. I'm not going to talk about his rates now, but I put this up now because geneticists see evolution happening very fast. And they don't believe it can be representative because they think they did it. This is my interpretation. I think geneticists don't believe what they observe in their labs to be anything that nature could do because, after all, they did it, not nature. And I ask you, if you can do it, do you imagine nature can? So I think there's a lot of real information in what geneticists find out in lab experiments. And I don't think we should dismiss it as something that they as a human did. I'll come back to the human secular trend too. How fast is evolution? Here's a diagram that I published in 1983, and I apologize because it's in Darwin's over here. I didn't know any better. And here's the measurement interval. This was literally an accident of plotting rates versus their denominator, time interval of the rate, the quotient, in other words, the ratio against its denominator, playing around with some data I'd compiled. It was on an old deck writer. Some of you might be old enough to remember that, and the paper comes up through the thing. And here came this incredible plot where these were perfectly inversely related. And that surprised me because I'll show you in a minute the plot I expected. I didn't know what the intercept would be over here, but I expected these would make some horizontal distribution somewhere on the page, and that would give us some idea about how fast evolution is. I had compiled the data from lab experiments, a few. I didn't have Falconer's data yet. Historical rates, which are things like knowing that rats were introduced on the Galapagos Islands probably 150 years ago now, or 200 years ago. And Jim Patton at Berkeley studied them on all the different islands and showed how different they are from island to island. And you can back out then what kind of a rate must be involved on a 200-year time scale. So those are plotted here. Then the rest of these are fossil rates from the fossil record, Pleistocene rates and Cenozoic rates, some from my work, but not many. And then older things, and these are some dinosaurs that were studied on a very long time scale. Well, what does it mean that we've got this perfectly inverse relationship? I told you rates are ratios. It means that here the numerator is highly constrained and the denominator is free to vary a lot. Now I could plot this better if I plotted the numerator versus the denominator, and you can see that the numerator doesn't change and the denominator does. Here I've plotted it as a rate, which yields an inverse relationship. And the reason is that this only varies by a few orders of magnitude up here, and the geological time over which evolution has taken place varies by, what, 10 to the 8th order, 10 to the 6th, 10 to the 8th orders of magnitude in terms of generations. Huge variation there, and much more constrained morphology is. Now, I got more flack than I could imagine. I mean, this luckily was published in Science, so people saw it, but Steve Gould immediately wrote a response to it about how this was a psychological, I'm still struggling with that one, Randy, and mathematical artifact of plotting a ratio against its denominator. Yeah, it's plotting a ratio against its denominator, but it's not an artifact, it's just an empirical plot. That's the pattern. And a colleague at Michigan, Fred Bookstein, got on a high horse and said that this proved that rates don't exist. Evolutionary rates don't exist. And I won't go into that too much here. I'll go on to the next slide to explain how he was thinking. And there is a sense in which he's right, but it's a kind of a narrow sense. The way Fred was thinking goes back to the original presentation of fractals. Fractals now have become kind of distracting, pretty pictures, but really initially they were about fractional geometry, fractional dimension. And the example given was a question, the title was, how long is the coast of Britain? Up here. Here is the west coast of Britain, Wales and Great Britain. You can see it's pretty complicated. And it turns out how long it is, this is Mandelbrot here published in Science 1967, how long it is depends completely on the scale of measurement. How long a ruler you get out to measure that coast, or what is the scale of the map on which you apply your ruler. It all depends on scale. And the shorter the ruler, the longer the coast. You pick up more of the detail. I mean, that's even intuitive. You pick up more of the detail, and so if you plot how long is the west coast of Britain, here are examples, data, that Mandelbrot actually worked out empirically, and you can see that there's a negative relationship on a log-log plot. So this is the proportion of the ruler, and this is the proportion of the length. And the length depends completely on the scale. Does that mean the length doesn't exist? Which was the conclusion Fred Bookstein was jumping to? No, it just means that to give a length, you have to also give the scale. For the length to be meaningful, you have to also know the scale. And so that's going to be true with evolutionary rates. They're going to be very different. They're going to decline just like this. We already saw that with that -1 slope in the previous plot. They're going to decline just like this over time. And the question is, what is an appropriate scale? Now we're doing rates per unit time. What is an appropriate unit time? I'll come back to that in just a second. I'll digress here by, you probably wonder what I do empirically to contribute to this. I study fossils through time. I study them in the Big Horn Basin in Wyoming, east of Yellowstone, down in the basin where, as the mountains around Yellowstone went up, the basin went down, sediment eroded, filled the basin over a long couple million years of time, flood by flood by flood. On that developed soil after soil after soil. Every soil had a forest and woodland on it. Every forest and woodland had animals living in it. The animals had teeth like we do. They're very hard. They preserve as fossils. And you can go out today and crawl around on these slopes and pick up the teeth. And if you record what bed they came from, you get plots like this one. And here's an example of something that didn't change much really through time. It zigs and zags some up through time. And there's an offset here that turned out to be very interesting because that's a global greenhouse warming event called the PETM, or the Paleocene-Eocene thermal maximum. It's a very good analogy for what we should be worried about today. And this has been studied. There are, I don't know, probably 1,000 papers written on the PETM now. But it was first seen in these excursions of dwarfed fossils in plots like this. Now, in terms of evolutionary rates, I'm interested here. The red line tracks the means level by level up through the plot. And so each one of those has an associated rate of change per unit time, and those are all plotted here. And I'll say a little more about that. So these are rates in standard deviations per generation. I've calibrated this in terms of the generation expected for a fossil this size, and here's the rate in terms of standard deviations per generation. Now, why standard deviations per generation? This is how evolution works. We in this room are a population. If we measure our stature and plot it as a histogram, it will look like one of these. Now, it will mostly look like this one because you're mostly young. Back in my day, it looked like this one. And when we talk about the human secular trend, we're talking about change from this generation to the next. And I've scaled this empirically. I mean, I've scaled this to fit what we know about measurements of human stature. This is my stature, the stature of my generation. The blue line is the stature, let's say, of your generation. I call this the professor or my generation and the student generation because I use this in teaching. And I really think I can see that the students I've taught for the last 40 years are taller now than they were when I started. Maybe I'm shrinking, I don't know, but I really feel like I can see that they're taller. And I'll show you a plot in a minute that this has been documented. This is happening. So that's what this is drawn to represent. Now the red line on the right up here is the sort of target that selection has to go for with 50% heritability to fall back to the blue line. So this is a model. But it shows the units that we're interested in. We're interested in how much of the spread of this normal distribution that we represent does the change encompass. And I've marked off here, there's the original mean, there's a +1 standard deviation, 2, 3, you know by the time you're at the third standard deviation, you're way out in the tail. Same thing on the negative side, so the whole range of virtually any, well, the whole range of any normal distribution is about ±=6, total 6 standard deviations. And what we find in human secular trend, like we find in study after study after study, is that the change here is about 1/10 of a standard deviation per generation on a time scale of 1 generation. So we're going from one to the next, 1/10 of a standard deviation. And if there's a number I want you to stick in your mind and carry away with you and think about, it's that number, 1/10 of a standard deviation per generation on a time scale of 1 generation. Now that might be different if you average over 10 generations or 100 generations or 1,000 generations, but the process takes place on a generational time scale. That's a key idea I want you to understand or dispute if you don't agree. Evolution, natural selection, doesn't care what's coming, doesn't know what happened in the past except as so far as that history is carried forward in terms of us now. And in the next generation, it will be the same thing over again. Us now. The next generation over again. Us now. It happens from one generation to the next over and over and over again. And yes, it's averaged over long intervals of time, geological time even, because the history of life is long. But what happens over geological time, Steve Gould, is not the process. The process is what we see today. And that process happened over and over again in the past. Okay, now coming back to my Wyoming mammal, here's an enlargement of the rate plot. And the thing I want you to see about this is here I have fitted a regression line of rate on the denominator, rate on time interval in generations, to the points. It isn't horizontal. The rates scale with the interval length. So these average anywhere from 10 to the third to 10 to the fifth generations in terms of interval length. They're lower on the long end and higher rates on the short end of the interval time scale. What do they tell us about evolution today? What do they tell us about evolution on a 1 generational time scale? Now this is a log axis, log 10 axis, so 10 to the 0 is 1. The intercept of this diagram is the rate on a 1 generational time scale. And here we are, here's a tenth of a standard deviation, here's a standard deviation per generation, and we're up here, I'll round it down a little and say a tenth of a standard deviation. That's what this extinct condylarth 50 million years ago tells us about evolution on a time scale up here of 1 generation. That's just what Douglas Falconer saw with his mouse experiments. That's just what my inverse plot that I showed you at the beginning of rates on a huge range of time scales show. A tenth of a standard deviation, more or less. Now, this was funny. A couple of years ago, I was preparing a version of this talk, and my sons were looking over my shoulder. They were both in high school taking calculus. And one of them said, "Dad, the first thing we learned in calculus is that average rates are meaningless." I thought, well, good, because that's exactly what I'm saying here. If we go back, if you take the average of this distribution of rates, that's what the blue line is. It would lead you to think that the process takes place at 10 to the minus fourth standard deviations per generation, when it's really about 10 to the minus 1. That's 3 orders of magnitude difference if you average. And that's what people do all the time. Once you catch on to it, it makes you nuts to see this averaging, because even high school kids are learning not to do that. So here, coming back to the original plot, this is what I published in Science with some more data added. It's the same 4 categories of evidence from lab experiments. Now I've got Falconer's in here. That's what this scatter is. Historical rates, there's some more. I haven't kept up too much with post-Pleistocene. There are a lot more fossil record rates in here. Here's what I observed, and to give you an image of what I expected it to be a plot that would look like this. I just didn't know where the intercept would be. So this plot might be down here. I didn't expect this inverse relationship. Now, taking a couple examples, here's the human secular trend. Now this is the one diagram where time is on the abscissa, and the stature through time is on the ordinate. And so this is an ordinary rate plot where the slope is the rate. And what does this come out to be? It's about a tenth of a standard deviation. It's about a centimeter per decade is how Townsend Bowles represented it. A centimeter per decade, well, that turns out to be, again, our old familiar tenth of a standard deviation per generation. So coming back to me speaking to my students, I'm not wrong to say that the change in their stature compared to my generation, their generation compared to my generation, could be evolution. It's not too fast. And when I say I think I can see it, that tells you something about evolution. We can almost see it. We can make ourselves think we see it. Now here's what I'm going to say about punctuated equilibrium. It's in 3 slides. This is the classic textbook example of punctuated equilibrium in the paleontological literature, at least. It's the best fossil example of punctuated equilibrium. It's in a bryozoan called Metrorhabdotus, published by Alan Cheatham in several successive studies. This is from 1967, which is kind of his big overview monograph at the end. And here I've taken one example. He has more, but they're all like this. Metrorhabdotus boldi is a species that he identifies that has the form that comes up through here. This is on a Melanobus D multivariate time scale. So what the numbers are doesn't matter. What we're interested in is how much difference there is between the D values. And the Metrorhabdotus here all stay pretty much the same from more than 7 million years ago to a little less than 4 million years ago. Okay? And there's another species, Metrorhabdotus codicei, that does this. And then we are asked to believe that the change from here to here could not be explained by the change from here to here. And, yes, I'll grant you, if you're talking about the long-term rate from here to here, that won't explain the change from here to here in the amount of time involved. But what about all the zigzag stuff going on in this sample, in this species through time? What do all those rates mean? There it's negative very fast, there it's slower, positive very fast, slower. Some of these are pretty high rates. So let's make a rate plot like I like to do, log rate versus log interval. Here are all the empirical rates in the blue lineage, the one on the left, bold eye. They scale with time, and they tell us on a time scale of 1 generation to expect a change of about 0.4 melanobus D units per generation. Now let's go back to the original plot and ask what that means. Here's our 0.4. If we start here and evolve at 0.4 melanobus D units per generation, we'll be off the chart here before even a fraction of a million years went by. If we say, okay, we're going to set this up at different rates and let it do a random walk to get from A to B, we can get from here to here by chance with a rate that's much smaller than what we observed. So don't let people compare rates on this time scale from here to here with rates on this time scale from here to here with rates on finer time scales. The time scale, the denominator, is important. So what does this mean in terms of punctuated equilibria? It means that punctuated patterns, at least the ones that we've seen documented empirically, can be explained perfectly well by ordinary Darwinian gradualism, generation after generation at the kind of rates we observe. There's no problem. Punctuated equilibria might be a valid pattern, and I'll say a little more about that now, but it is not something categorically different from Darwinian gradualism. It can be explained perfectly well by gradualism through time if you understand the rates involved. Now, why do we get punctuated patterns? I'll grant you this one stays pretty much the same, and this one comes in here. We don't know quite where it came from. We think it came from here. Cheatham thought it came from here, and stays pretty much the same. Why does that happen? It has a whole lot to do with the history of the Earth and the history of the environment, and what happens if you have these high rates that I'm talking about and you start an evolution experiment with lots of species diversifying. They're going to fill the space available very quickly. And I like to give the example using mammals. Mammals can't be smaller than a least shrew, about 3 grams weight. They can't be smaller. They can't maintain their body temperature at a smaller size. They can't be bigger than a great blue whale in the sea and something smaller like an elephant on land. They can't be bigger than that. Those are only separated by 3 orders of magnitude. In the time since they diverged from each other, a great blue whale and a least shrew, is on the order of 7 orders of magnitude generations back in time. Now think about that. That's like diversifying. I'm a geologist, so indulge me here. That's like looking down an oil well with a pipe 10 centimeters across, a kilometer deep, and seeing that evolution, starting at the beginning, filled that pipe in the first 100,000 years. And where's it going to go after that? The pipe is full. Everything has got every species has its niche relative to everybody else. There's no place to go. So what happens? Nothing. They just keep living. But what if you perturb the system sometime? What if you have a greenhouse warming event like I showed you at the beginning of the Eocene? The system's perturbed and it starts over again. And it has to fill that pipe again. Everything isn't gone. There are things there to found the subsequent generations. Punctuated patterns, the basis of punctuated equilibria in the fossil record, are a record of the perturbations of life up through time, not something about how evolution takes place per se. They're a representation of the history of life and not of the evolution of life. So I have no problem with punctuated patterns, and I have no problem explaining them as Darwin could with gradual evolution. Now I'll give you a few more examples where this is important. Here's a study. This is from the paper I suggested as reading. It's really from a study by Michael Lynch, where he was trying to document rates of polygenic mutation. And I worked out one of the examples starting with the original literature. Here's a mouse experiment where mice were highly inbred to make them uniform and then allowed to diverge randomly to get some sense of what polygenic mutation was doing in terms of morphology. Now, if you come back after one generation, you can't measure anything that's happened. 10 generations, it's tough. So in this case, the mice were measured again, at least reported, to be measured again after 18 generations. And these blue dots are the results of several different mouse lines allowed to evolve like this. And here they came back after 93 generations of divergence, and here are 4 lines that survived. And Lynch, like everybody else would do, took these numbers and these numbers, here's one down here, and averaged them and says in a table or in the text that the rate is about 10 to the minus 2 something, what is that, mutations per generation. But look at this. These are here, and these are here. Here's a regression line fit to them. If you want to predict what these tell you about what happens on a one-generational time scale, it's here, not here. And that's at least an order of magnitude different. So be careful. Don't just average this stuff. Think about how it scales with the time scale of what you're studying. Here's another example. This is from a great study by Vincent DeNeff, who's now at Michigan, and Gillian Banfield at Berkeley. DeNeff was at Berkeley as a postdoc with her. And this is a paper published in Science in 2012. As soon as I saw that they had these data, I thought, oh, I wonder, I'll bet that scales inversely like I expect. And sure enough, when you plot the rates over here, log rate versus log interval, here are their points. Plot a regression through it. Here's what it tells you about evolution on a time scale of one generation, the time scale of the process. They averaged them and reported this. That's about 2 orders of magnitude difference, underestimating what's going on. Now this is important because people routinely take data like this, calibrated back in time, and use it as a molecular clock. Here's the calibration. Here's the average rate right through the middle of the points. So if we see this much divergence, it must have been this much time. If we see this much divergence, it must have been this much time. There's your molecular clock. But the points themselves tell you that they're scaling with time like this. So anything you infer about time in here, yeah, if you infer something here, you'll get it right. Anything inside you infer younger than that, the rate and the change will be underestimated, and the inferred divergence times will be too long. Oh, sorry, it will be too young. It will be too short. Anything out here, rates and change are overestimated, and the inferred divergence times are too old. And guess what? As a paleontologist, I can tell you that molecular clocks systematically underestimate short, near interval rates and overestimate the long ones. So we need to think a little more critically about just throwing a line through here, an average, taking an average and imagining that that's the line that the points represent. I won't belabor that, but it's pretty important. Okay, now what about random walks? Are a very common model in evolution. And here again, here's an envelope of 1,000 or something, of 100 random walks. That's what the gray lines are. Here's one example. And then the blue is the envelope of all 100, or the expected envelope of all 100. We could do 1,000 or more, where we expect them to fall. And so if you want to know what's the limit to rates on this time scale, it will be different from what's the limit to rates on this time scale to what's the limit of rates on this time scale. And just to hammer that home, here are all those rates on all the time scales plotted. Now there's so many I could only plot every 10th one. But even so, there are how many rates? So there were 5 million rates come out of that little experiment, and I plotted 500,000 of them. And here I've plotted the rates, the median rate on the for each time scale, and that's what it looks like. So if you take the average rate for all those rates, you'll get something down here, an order of magnitude and a half less than what you know, because you created the model, you did the experiment, you know is the answer. That's the rate that generated all those curves. Now, you know, we as evolutionary biologists aren't the only ones who suffer from this problem of averaging rates. Here are rates of glacial movement in Switzerland, I think, I forget, published in Science in 2012. And here are all the rates for one of these glaciers. I don't remember which one it is here. It's one of these. All the rates that are known for its movement, and I think the black ones are downhill and the white ones are retreating or something. And the average of those that's reported is 4.3. What does that mean, 4.3? Well, the average comes out to be here 10 to the minus 0.4, something like that. Okay, that's 10 to the 0. What? Anyway, the difference is about an order of magnitude and a half less is what was reported than what the data really scale to show you about what's happening on a per year time scale in the case of glaciers. I'm imagining they grow seasonally and retreat seasonally, and so a year would be an appropriate time scale, I think. So here's an example where this works too. Now here's another one to think about. Extinction rates are really difficult to study because about all we can do is ask, how long was a species here before it became extinct? And then the rate is just one species was here for a certain amount of time and it became extinct. That's one extinction per that amount of time. And so they are necessarily inverse curves like this. Now here they've turned the time scale around from the way I would do it. I would plot it so that they were declining with longer time scales. Here they've got the longer time scales on the left side and the shorter time scales here. And then they argue, this is in these hot colors and everything because it was published in Nature and got a lot of press. But here are the extinction rates of things calibrated today. And here are the fossils that they're compared with. Now, the first thing that puzzled me, and I still don't understand it, is why these are not inversely related to their interval length. To me, that says they're not calculated right. But I haven't gotten into that. Somebody else needs to. But I want to ask you, if this is what's happening on these shorter time scales up here, and these are longer time scales, what does that tell you about current extinctions compared to the fossil record on these time scales? The current ones aren't higher, they're lower. You follow me? If this is how the extinction rates scale with time, inversely like I talked about, then when you get down to the kind of time scales for which that the fossils represent, they're higher, not lower. So this has just been misinterpreted. I don't know what the answer is, because there's really nothing to do with the fossils except what we've done here, which is divide them by their duration. And where are they going to fall? They're going to fall on this line farther down. So extinction rates are tough to compare. Okay, conclusions. Geologists, paleontologists, biologists are accustomed to long intervals and low rates that are observable. We studied 10 generations, 90 generations because we can observe change after that amount of time. Well, I don't know why we don't try on shorter time scales, except with small things they're hard to measure. They're rarely representative of the process of interest. Rates on the order of 1/10 of a standard deviation per generation on a time scale of 1 generation. That's what this gen.gen means. Or what I called 1/10 of a Haldane is what we see over and over again on a generational time scale, which is the time scale of the process. This is very fast. This means you can cross a normal distribution in 60 generations. Cross the whole thing. That means you can go from being averaging the shortest, one of the shortest people we can find, to one of the tallest we can find in 60 generations. Now, it's still, it's not overnight, but it's not geological. So many processes are much faster than commonly recognized. Even glacial advance and retreat are faster than represented by averaging. And in terms of extinction and conservation, our credibility depends on getting these extinction rates right, and I don't know how to do it yet, but it's worth working on. Okay, I've left a little time. I probably should ask a generic question since my host is focused on evolutionary medicine, and I think I'll turn that around and ask you, what does this have to do with evolutionary medicine, or at least ask you to think about it. Thank you.
I'm going to very briefly answer Philip's question before I let you know some more questions. There are two things I get from doctors when I talk to them about evolution. The first thing I get is, hasn't evolution stopped?
Yeah, that's important.
That's fairly bizarre. Another thing I get, and even some other evolutionist psychology types, and others say, nothing's changed in 10,000 years. We Stone Age, no, wrong. And then the other thing I get is, why do we have genes that cause nearsightedness? And a lot of doctors believe it's because glasses were invented, relaxing selection on those genes and drift could make those genes drift to prevalence to cause 30% of us to be severely nearsighted, which of course is preposterous. So I'm hoping doctors can pay attention to at least the broader perspectives here, but I bet there are a lot of technical and other questions for your talk. Let's see what people want to say. I'll let you call on people, Philip.
Okay, yes, here.
Do you think this, like 1 standard deviation per generation, is like an average rate or like a cap on the rate? Do you think there is like a maximum rate of evolutionary change?
Well, let's say that it's a typical rate. It's, you know, if you look at Falconer's mouse experiments, there's a huge range there, and 1/10 of a standard deviation is in the middle. So there are faster rates recorded. Now, you can't, I didn't talk about that double histogram, double bell-shaped curve example quite the way I should have, because that's actually scaled with a certain gradient of selection. And the upper shift is a result of that gradient. And you cannot have a response. Turns out that if you're talking about gradient selection, the response asymptotes, and so you cannot get above, I forget the number now, something like 0.4 standard deviations per generation. You can't get above that. But of course, gradient selection isn't the only selection. You can have truncation selection, where if you truncate the whole distribution, you know, you're going to have one survivor maybe very different, but an inviable population. So there's a trade-off there. But 1/10 of a standard deviation comes up over and over again. Yeah.
During your talk, you use words simple, fast, quantity, rates, measurements. Now, as an engineer, if I can measure something, I can plan. And in Dr. Nessie's book, While We Get Sick, I remembered a line in there that interests me. And he says, no plans contribute to evolution, and there to be no fresh starts. Evolution consists entirely of trial and error teamwork. So based on what I just gleaned from your lecture today, can evolution plan?
Okay, well, let's go back to the beginning. I should be very careful because we cannot measure rates. We quantify rates. We measure difference. And these are statistical shifts, and I'm saying typically we see 1/10 of a standard deviation. So I'm saying that we can plan in the sense that we can expect that, but that all depends on on the selective regime, and we don't know that from generation to generation. So that's a typical thing we see in the lineages we sample. I don't think we can use it for prediction. I mean, we can use things like at least true at 3 grams in a great blue whale at 100 metric tons as limits on where mammals can be. Those are probably more or less physiologically fixed. But where we are in that depends on, and how we change in that depends on what is the selective regime is every generation, and every generation it can be different. So yeah, planning's not going to help us much. Yeah.
Well, there's a field that claims that we can understand and predict everything about current modern human behavior because essentially we have Pleistocene brains. That field is called evolutionary psychology.
Mm-hmm.
And they say, the adherents to this dogma say, well, the current brain has to be a brain that evolved in the Pleistocene.
Mm-hmm.
Why? Because there hasn't been enough time for really significant brain
Mm-hmm.
change. And besides, the cognitive modules are complex, and the evolution of complex characters is really slow. So I want to just quiz you about this word complex.
And do please repeat the question for the camera, please.
Complex.
Philip, not you, Kim, Philip.
All right.
Okay.
So my question is, if I were to throw in that word complex and say the brain can't have changed very much because the adaptive modules are complex, do you think that would change anything about what you just said?
Well, it's an empirical question, and the question is, do we have Pleistocene brains that are not capable of change in what? In
10,000 years, what is that?
yeah, in 10,000 years or something.
400 generations?
I mean, I hope I've answered that question. There is nothing about complexity that doesn't have a variance. If you can quantify that variance, and It might be a very narrow bell-shaped curve, or it might be a very broad one. But the rule of 1/10 of a standard deviation will be relative to what the variance is in the trait you're interested to measure. And so you have to be able to quantify it to know what the answer would be. Your question reminds me of something I read in Cosmo magazine 20 years ago. And I read it because it was an interview with Steve Gould. And he insisted that human evolution is over. It has stopped. And that's just nonsense. You know, we can't get away from it. It's part of nature. And that's true of your psychology colleagues here.
So I'm sure there are lots of more good questions, but we're going to stop. It's 1 o'clock. Thanks very much.